Namaste students. I am Jivan Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic size exclusion chromatography, which is the part of the separation by the chromatographic separation method. So in this lecture, I will discuss about the basic principle, basic mechanism of size exchange chromatography. So exclusion chromatography is also known as the gel permission, gel filtration chromatography. It is also known as the GPC. Okay? And separate molecules according to their size, uh, low resolution, and quaternary structure. So that is the basic of the size exchange chromatography. Molecules will be separated on the basis of their size. What are the actually examples of size exclusion chromatography? We have understood in previous lecture that thin layer chromatography and from paper chromatography are very important techniques. What are the basic of the thin layer chromatography in the paper chromatography? In thin layer chromatography, we were using a stationary phase and a mobile phase. In thin layer chromatography, the separation was conducted on the basis of the size also. Not not only the size, but there was a part in which it was separated on the basis of the size. So similar, not all the things are similar, but similarity will be found in the size exclusion chromatography. The example of this size exclusion chromatography is the gel electrophoresis, in which we separate our particles on the basis of their size, and we separate DNA molecules, protein molecules. So this is the example of the size exclusion chromatography. So discuss about the size exclusion chromatography. What are the basics of this technique? In size exclusion chromatography, the first thing is that stationary phase. Okay, what is the stationary phase? The stationary phase in size exclusion chromatography is beads. Okay, there are some beads which have some hole, which have particular pore size. Okay? They have a particular pore size, and the examples of these beads include the hydrocarbon like dextran. So dextran can be used as a stationary phase in the size exclusion chromatography. The mobile phase, which which is used in the size exclusion chromatography, is the liquid. It is a liquid, and it might be any organic solvent like petroleum ether, acetic acid, whatever we can use. So it can be any organic solvent which we can use for the separation of the particles. So this mobile phase, the solvent will carry our sample along with it, and the separation will be conducted. What is the characteristic of separation in the size exclusion chromatography? The separation in the size exclusion chromatography is conducted by size. Means the small particles and the large particles. Okay, the size of the molecules are two size: small particles and large particles, and they have particular size. The small particles will be able to move faster. Okay, they have a ability to move faster, and they can be passed through the beads because they have lower size. They have lower pore size. Okay, and the larger particles will not be able to move faster because they have larger size. So this is the characteristic of the separation of the size separation and the size exclusion chromatography. What is the mechanism of separation in the size exclusion chromatography? The separation is conducted on size. It is based on the size of proteins, which can pass through the holes in weeds. As a result, they pass in the column first and then vice versa. They will pass the column or they will not be able to pass. So on that basis, they are divided. Talk about the mechanism of size exclusion uh, chromatography in detail. So what is the basic of suppression? Uh, Talk about the mechanism. Then suppose that there is a flask. Okay, there is a flask in which we have some beads. Okay, there is a flask in which we have inserted many beads, and these beads have a particular size, and we have inserted our sample there on over the we have inserted our sample over the glass beads. Sorry, beads means the beads are composed of the dextran. Okay, we have inserted our sample over the dextran beads. Then we add we add solvent. Okay, we add solvent. So what actually happens? The molecule which are on the upper side, so they will come in the bottom side along with the solvent. So solvent are the mobile phase and the beads are stationary phase. So the solvent will carry the sample. Okay, they will carry the sample according to their polarity towards the bottom side, so that they can be separated by a wall. So how the protein, how the products will be moved, they will how they will move. So if we know that what is the size of the dextran beads, they are the size of dextran is known. So if the molecules which are larger than the dextran size, they will not move easily. Okay, they will not be able to move easily, and the molecules which are smaller than the dextran, they will be moved very easily. We can also say that the thing which support the movement is solubility with the organic solvent. The soluble compounds, okay, will will be able to move faster, and they will move towards the bottom easily, and insoluble will not be able to move, so they are not soluble in the mobile phase. They are not soluble in the organic solvent, so their movement is very hard. So this is the basic of side exclusion chromatography. I must share the screen here so that you can understand. So this is the diagram. This is the porous polymer beads. Okay, so these are the porous polymer beads, and it is our protein mixture. So first we add our protein mixture along with the solvent. So they are added, and how they will be separated. This is the wall on which the protein molecules will be separated. So when we have added our protein molecules in our beads, then the protein which are larger inside, okay, they will separate it. They will migrate towards the end very freely. Here we can observe here. So it is really opposite to it is opposite to thin layer chromatography in the paper chromatography, in which the smaller particles were being separated uh, easily. In this, the larger particles are being separated freely, so they will move inside. They will move towards the uh, they will move towards the end very freely because the beads will capture here the beads will capture all the small particles. Okay, so there will be space for the large particles. The small proteins will be captured by the beads. Okay. Their pore size, the small pore size of the uh, small particles is smaller. Okay, so they will bind to the beads, and the molecules which are uh, larger in size, they will not move towards the beads. Okay, they they have particle size which is not uh, smaller than the beads, so they will not move towards the beads, so they will diffuse freely. So the which first product which will be separated in the size exclusion chromatography is the large proteins, because they will not diffuse inside they will not diffuse inside the dextran beads, so they are free to move. Okay, they are very free to move, so they will be separated easily. But the small particles will be captured by the dextran beads, so their separation will be in different beads. So the large and small particles will be separated uh, in different ways here, because of the uh, size of the proteins, and large size of the proteins, they will be separated freely in the fraction in different fractions, and the smaller particles will not be able to diffuse because they are present in the beads. So at the time of the separation, uh, we are observing in the tube number one, there is no, if it was collected in the tube number one, and there was not any kind of color in the tube number two, we are observing that there is no color. And when it is color, then we separate the particle in a tube number three, and when the color changes, we separate the product in a new tube, and when it will also another time it will change, then we separate it in another tube, so different fractions will be collected from this size exclusion chromatography. So this is the basic of the size exclusion chromatography, in which the beads will capture all the small particles. Here they will capture all the small particles, and the large particles here will move through the walls. They will be move through the walls, and the small particles will move through beads. So they will move through the beads, and the large particles will move freely. They will not come inside the beads. So I hope the students will better understand all things very well. So in this lecture, I explained some basics of the size exclusion chromatography with explaining the mobile phase and the stationary phase, and how the mechanism is used for the separation. What the mechanism is used for the separation of the particles on the basis of their size. So I hope that you have already understood all these things. So that mm, the question which can arise, the examiner which can ask, I explain size exclusion chromatography with their basic principle and their uh, general mechanism of by which the particles are separated. So you can answer it very easily. So my good wishes with you, and I hope that you will be better to answer all these questions. So my lecture is over here. If you have any questions, you can ask me. Thank you very much.